All right, let's go ahead and get started. Thank you for coming. It's a great pleasure for me to introduce Professor Daniel Cohen from Princeton University. And he is currently assistant professor in the Department of Mechanical and Aerospace Engineering. He received his bachelor's degree in mechanical engineering from Princeton, and then went on to do a bioengineering joint program from UC Berkeley, UCSF. We're working with Mikhail Marovitz and working on Galvanotaxis and collective cell migration. He then went on to do a postdoc with James Nelson, who's a very well-known scientist in looking at cell interactions in the context of complex tissues. He was actually the last postdoc with James Nelson, which is very interesting. He started his lab in 2018 at Princeton, and he's developed a very interesting program, in my opinion. He has very wide range of research interests, looking at organismal biomechanics, from dinosaur to even tardigrades to actually looking at collective cell migration. So I met Daniel just a few months ago, and I was really captivated by his presentation. So I thought I should invite him to give a seminar in our department. Outside of lab, he does actually science communication for the public, and it is really intriguing to hear him talk about the history of science and in various areas. And I think it's going to be something that he will touch on hopefully today. So without further ado, Daniel, the floor is yours.
Thank you.
Yeah.
It's very exciting to get to be in Michigan because I've never actually been here and I appreciate it. And my only experience of University of Michigan was in that horrible movie, whose name I can't remember, with the guy from How I Met Your Mother. Anyone know what I'm talking about? It's terrible.
Yeah, it's not good.
So,
Zingerman's was in it.
okay, well, it's better in person. I can tell you that much. So thank you for making the time. And we'll get started. So let's see if the movies are playing. Okay. So as Alan amazingly remembered, I went through mechanical engineering, bioengineering PhD in an electrical engineering lab and a cell bio postdoc. I don't know what I am anymore, but I had been working on nanotube electronics, inkjet printing, and dinosaur mechanics and lizard mechanics and things like that until I had a chance to do this experiment. And so I was at the Woods Hole physiology course and I made an epithelial tissue. So this is kidney tissue, blue nuclei, red junctions, and I cut it down the middle and the thing started to go like this overnight. And this was on an automated microscope, but I'd never seen anything like it. I stayed up all night watching it, even though it was recorded, because it was really cool. And so as an engineer, this is a pretty neat system from a materials perspective, right? Sure, it's a self-healing material, but there are lots of those. This is an agent-based material. This is one where every component of the material computes and interacts with its neighbors in an agent-based fashion that gives rise to complex behaviors that solve problems, right? Self-healing materials are just materials that handle a perturbation. This thing can detect different states and respond to them accordingly. We can't build those. This is pretty neat. So how does this work and what can we do with it are some of the questions that motivated the lab when we got started. And one of the frameworks that we think about for any collective system is this old concept called the Zellenstadt by Rudolf Virchow, the founder of pathology as a field. So he wrote a whole book called the Zellenstadt, it means cell state in German. And he tried to explain, you may even know this paper, he tried to explain his political theories using tissues. And he explained that organs and organisms are kind of like nation states and cities, and they have a whole hierarchy of organization inside them. They have waste collection. It's just like urban life. And he has this beautiful quote that the body's a cell state and every cell is a citizen. And he was trying to kind of be anti-fascist and he go against Bismarck at the time. And he got into a duel with Otto von Bismarck with sausages at one point, which you should ask me about later. But he really laid the groundwork for this concept of thinking about tissues as socially dynamic systems. And this is a lot of the tools that we use today, right? All the statistical mechanics from physics describe ensemble behaviors, urban planning, traffic analyses. Those are the sources where we come up with approaches to look at complex systems with lots of things interacting, right? So there's this long history of tools to work with these systems that we now get to play with. And what the lab thinks about is, okay, can we understand these processes enough to do something useful with them? Not just explain them, but build something or heal something more effectively, that sort of thing. And so, sorry, there are three things we'll go over today with respect to that. Some of our work just trying to think about cells as collective agents. some of our work on actively herding them, and some work on trying to mess with large cellular collectives that is not really related to the other two, but I want to show you the story. So we're going to be going over a lot of different ground. I apologize in advance. Hopefully there's something for everybody. Okay. So we'll skip over that. Come on. No. Okay. So just so that everybody's on the same page, I want to just explain how we think about collective behaviors, the basic rules, and then that'll set the stage for everything else. And because I don't know how many mechanical engineers are watching who are not cell familiar, we'll do a quick summary of cells too. But you got your single cell here, and it's already quite complicated. It has its nucleus for computation. It has actuators in green, the motor proteins, and then it has end effectors, which are the little feet here. So far, so good. Beautiful quote from John Bonner's book, First Signals on the Origins of Multicellularity, saying that even before multicellular things started forming, the cell was already so complicated, it had already lost its innocence, right? These are sophisticated little agents. But the real magic for us happens when you get more than one of them together. And boom, suddenly you get cell-cell junctions. That's the cadherin proteins locking cells together. They do not appear in single cells. So you can only start to get this complexity when you have multiple cells. And you get the beautiful kind of force balancing, the focal adhesions concentrate at the edges, and mechanical information transmits across the junction. And as many of you already know, when you get enough of those together, you wind up with this kind of classic sorts of self-organizing behaviors. And you only need 3 rules to think about those. The first, or sorry, before we get into those, in case you are not aware, all of this collective migration is central to us being here today, right? Embryonic development, large numbers of cells got to move all over the place, coordinate decision-making. Wound healing, large numbers of cells got to do the same thing. Cancer invasion, unfortunately, it's increasingly clear that there's a collective component to cancer invasion beyond just pure single cell metastasis. So all of these processes derive from these emergent behaviors in crowd dynamics. And the 3 rules that you need to keep in mind then are as follows. So this is kidney cell healing again. Local interactions create global order, meaning that if I interact with my nearest neighbors in a group and that's it, that behavior will propagate and produce a complex group dynamic without any shared information long range per se. The second is that this system is greater than the sum of its parts, which sounds like a cliche, and it is for a reason, which is that let's say we took a single cell out of this population, or a single bird out of a flock, or a single student out of a party. The dynamics would be totally different, right? In the absence of neighbors, you wouldn't even get cell-cell adhesion proteins. You would not know those happen. So no matter how closely you analyze a single bird or a single cell, you would never know what it would do in a group. You have to study these at a systems level. And that is nice because unless you really need the deep cell biology and pathway analysis, you're actually operating at a higher level. You're operating at the system dynamics level, the materials level. And so what you have to work with is a little bit different than pure protein engineering or protein pathways, right? You need to think about all the interactions. And lastly, nobody tells this system what to do, right? It heals on its own and it doesn't need our help. On the other hand, these systems aren't optimized. So it's a mistake to think any crowd behavior is optimal by default. Wound healing occurs, but it doesn't occur all the time or as quickly as it could. Scar formation's a problem, diabetic ulcers are problems, right? So maybe we could accelerate those processes or modulate them in useful ways. That's where the engineering part starts to come in. So the first question that my lab did, which was really just the, oh my goodness, the doors have just opened, we have all this new equipment and nobody in my lab knows what a cell is except me, was to say, what happens, let's take a stupid simple experiment. Let's grow millimeter-sized tissues and 3 or 4 millimeter-sized tissues. And let's ask only which one grows effectively faster and what are the growth dynamics. This was supposed to teach students who've never seen cells what cells were so that we could do the real work. And this turned into the first lab paper because it was super weird. So what we see collectively is this. These are MDCK kidney cells. They love to work collectively. The color is cell cycle. G2 are actively dividing cells. G1, or sorry, green is actively dividing cells. Purple is not dividing cells. And when these tissues grow, this is what happens. And they started at the same density. So solely as an effect of the geometry, you get what we call the watermelon effect here and the vortex here. The watermelon effect is easy to explain. The inside of the tissue divides too quickly, cell density goes way up and cells have to shut down division or they become a tumor. This is how most of your epithelia in the body work, like the surface of your eye, lining of the gut, the skin. When things get too dense, they shut down cell cycling, and that's purple. Whereas the outside stays green and continually growing. In the small tissue, it expands so quickly relative to the center that there's actually a density reduction briefly, which triggers a force imbalance that essentially leads to vorticity. And these vortices will spin for days. It's pretty surprising. Now, what's important about the central question really is just that actually they both grow at exactly the same outside speed. Don't pay too much attention to all the plots. I'm just showing you we did math on these, we understand them. So they grow regardless of whether you're small or large. Your radial speed, which, come on clicker, is this yellow band here, is identical. They grow at the same outward speed. The reason a small tissue grows faster than a large tissue then is just perimeter to area ratio, right? Small changes in area are more impactful in a small tissue. And that drives all this internal decoupling because you can see the insides of these tissues look different. And that's what gives rise to vorticity. And these are orientation maps just showing the strength, location, and power of these vortices in small tissues. And what's important isn't really that these behaviors happen per se, it's that they lead to a bizarre conclusion, which is that, okay, you have constant edge speed, fine. The outside is totally decoupled from the inside, fine. But what it means is how you get from A to B matters. So suppose you take a 1 millimeter tissue and you grow it to 3 millimeters, and then you compare it to a tissue that you made starting at 3 millimeters by pouring the cells into a mold at 3 millimeters to begin with, right? Totally different behaviors, even if the densities are matched, because the mechanical history of the tissue that grew up changes its life behavior and trajectory, right? All the forces are now coordinated in a different way. The cell cycle is different. Everything about that tissue is different, even though it looks the same as the other one. And no one knows what's better, but in tissue engineering, we never actually talk about the best way to grow a tissue, right? There's an assumption that you need to just get the cells in. And the question of, do you let them grow up a little bit, or do you want to seed them at the negative density is unclear. And it matters. And so that's just something that we took away from this study that's actually kind of surprising. Now, as always happens with time lapse, somebody doesn't want to take things off the microscope because the experiment ends in the middle of the night, right? So then you wind up with fun things where you let things grow too long and they collide with each other. And that started a second study that we just wrapped up. So here's your classic thing where you have two tissues. Now the color is not cell cycle, it's just membrane dye now. And if they collide with each other overnight, you get your classic zipping here. But if you have asymmetric tissues, you start to see really strange boundary dynamics. And they're extremely stereotypical. So in that first paper, we actually used active fluids to come up with a computational model that predicts the internal dynamics of these tissues as well as the growth. And we fit that model to these. And now we have a computational tool that can pretty much completely predict how any arbitrary pattern of epithelia are going to grow and interact with each other. And that all depends on the local density at the interface. So you can check in the paper itself. We have a nice series of movies that shows how dense tissues versus low density tissues push each other out of the way and things like that. But it all just comes down to wherever you're locally denser, those cells will push on the other side until the density gradient balances out. And that produces really predictable things. And so skipping ahead to some of the implications of that, I asked the students to make a pretty picture because you always need a pretty picture for these types of articles. And so we said, let's make an Escher drawing. And this is called a dice lattice. It's one of Escher's sort of cuboidal structures. And we said, how do you make this with tissues? Well, you need 3 different colors, okay. And we ran our simulator and predicted that the dark outlines would produce this structure. And so we created that here and then let it run for 96 hours and it produces that pattern as predicted over time. And this is a high integrity. You can use a special Petri dish that lets you pull a tissue up intact, and you can pick this tissue up out of the dish and move it somewhere else, put it down. So now you have a composite with 3 different types of tissues in it. You could put inducible promoters in them and turn on different genes and signaling across it and grow all sorts of complicated things. You can look at force balances. It's a neat system just to prove that you can engineer the collective behaviors using intertissue collective dynamics. We had a reviewer tell us to take this out of the paper because it was nothing other than a shiny object. And that it was a disservice to the beautiful work of MC Escher to say this had anything to do with him. We did not take it out of the paper. because I think it shows that you can build things with it, which people often forget is an important thing to do. Some weird things that come out of this that are always fun is when the models get broken. This we call the Temple of Doom configuration. The purple tissue is going to try to escape as it grows. The green tissues are going to try to close it off. And what happens is, this is much later in the process. You're looking right at where the green tissues and the purple are racing. And you see this odd behavior where the purple tissue gets extruded down to a single cell-wide line. And the reason this happens is you have a non-traditional boundary. It's not two perpendicular or parallel boundaries. You have boundaries that are dynamically changing. The forces are no longer parallel to each other or anti-parallel. You have extrusive forces that pull on this. And what's actually happening is the purple tissue is going faster than the green tissues, even though they're all the same cell type. If you just make the green tissue by itself with the same curvature, you don't get this acceleration effect. So something is changing the signaling and migration right at the edge there and producing these really neat, almost developmental motifs just by tweaking the geometry. So again, you can get really fine gradations, and this may tell us something about development or how to build weird morphogenetic type systems. So we understand that stuff. We can build these things. Great. Can we poke them now and prod them and make them do something useful? This was genuinely inspired by me as a graduate student watching too much YouTube about sheep herding. And this is beautiful. This is contraction expansion dynamics. You have liquid crystal pneumatic ordering. You have a jamming transition. You have flow patterns. The dynamics of this are mathematically identical to human umbilical vein endothelial cells. That's provable. It's pretty cool. And what's more cool is we've been doing this for 7,500 years. And there are papers written every year on how sheep herding works. And the funny thing is the dogs, the shepherds, and the sheep do not read the literature. So how did they do it? The answer is, this is the power of swarm engineering. You don't need a reductionist analysis or control theorem. You need a perturbation control, right? If I move the dog here, the flock moves here. So then I move the dog here, the flock moves here. You can iteratively update your control to produce an outcome that's useful without fully understanding what the sheep-sheep communication is. Does that make sense? So now you're operating in this gray area of swarm dynamics in a living system and trying to see what you can do with it. And what would be nice is, this was produced by DALL-E. I should credit the AI for making cool art. This is, you can maybe heal injuries faster. You could alter morphogenetic processes. You might be able to control disease, so cancer invasion, or modulate inflammation if you can herd the swarming of neutrophils, things like that, and just grow tissues in new ways. And the problem is that we want to do this, and we have a system like this. Again, these are the classic collective behaving cells. And so to set some design constraints on this, we said, okay, whatever we're going to do needs to be interactive. Micropattern substrates are very powerful, like grooves and lines and things for guiding cell behavior, but they're non-dynamic, right? They're static structures. You cannot change how the cells behave once they hit them. We want something programmable. Second, we want something that takes into account the naturally occurring collective behaviors first, that works across a wide variety of cell types. We are doing some really cool optogenetic stuff with Jared Tocher's lab, but you've got to genetically modify the cells to do that ahead of time. So what we're asking is, can we take what's there and sort of harmonize with it and get some nice result out before we mess with anything else? So we look at chemical cues. Most people are familiar with chemotaxis, directed migration in a chemical gradient. The problem is not that many cells actually chemotax. It's surprising, but it's not as common as you would think. It's also very difficult to program chemotaxis, especially on short time scales and length scales because of diffusion. So we couldn't do that. Mechanical cues, I mean, mechanical engineer, it'd be nice if we could use them, really hard to program again. And there's no clear ability to really structure these things, although it's getting there. We know that if you align principal stresses and things, you manipulate division and stuff like that, but it's hard to build an interface around that yet. Civil engineering, we skipped over. Financial engineering, we skipped over. Electrical engineering, we're very good at electrical cues, right? We're probably more advanced at finely controlling electron flow than pretty much anything else, industrially speaking, in that sense. But there's this problem with electrical cues, which is that many of you probably have this image in your head, and you're not wrong. I will give you a very short summary of why people think the way they do about bioelectricity, and then we'll get onto some science again. But there is a long and sordid past to bioelectricity. If you do not trust it, good, you should not trust it. There were bad things that were done in the name of bioelectricity that go all the way back to when Galvani did his frog leg twitching experiments, which everyone's heard of in the late 1700s, it's like 1795 or so. People got excited, nobody understood it. Galvani's nephew is famous for going to London, getting recently executed criminals and electrifying them with voltaic batteries such that the corpses sat up and waved their arms and winked. And those were called the corpse galvanism experiments. They were theatrical events. People paid money. This is why he did this, to watch an executed criminal seem to be alive. He also probably bribed the court to get the bodies exactly when he needed them, and that's a separate story. And so already there's this weird, not so great feeling around this, as well as a fascination, right, from people. Frankenstein comes 10 years after the corpse galvanism experiments. This is the best adaptation of Frankenstein. If you have not seen Young Frankenstein, you should go watch that. The other version's not so good. And Frankenstein doesn't even mention bioelectricity anywhere other than lightning striking a tree in the first chapter. In Frankenstein, the technology is not important. It could be CRISPR or nuclear power, whatever it is, right? It's whatever the most advanced concept of the age is. Nobody needed to be told that because of these experiments. So everyone just assumed this. And within not that long, 5 to 10 years of Frankenstein, you have a mass of quack medicine, all based on bioelectricity. This is my favorite. These were called suspensories. They were all over the place in the 1800s. They are literally bioelectric underwear for men and women. They stimulate you down there, and they are associated with curing literally everything. I will read to you. This will cure all rheumatic complaints, lumbago, general and nervous debility, and wasting diseases of the body caused by indiscretions in youth, age, married or single life. So anything you do wrong, this bioelectric underwear will fix you. Obviously, it does not fix you. And this and everything else like it, like the chairs that statically charge your hair and cure your cancer, et cetera, do not give people a good taste and really start to backfire and poison the well, so to speak, for bioelectricity. With the result being that bioelectricity was actually banned as a research area in the US in 1906 by Abraham Flexner, who created the Institute for Advanced Studies at Princeton, but also reformed the American medical system and banned homeopathy and bioelectricity. There is a reason why, even though the phenomenon I'm going to tell you about was discovered well before chemotaxis, it is far less understood than chemotaxis. So going on in the background was a failure of science communication, but some good science. This is Dubois-Raymond. He started the field of electrophysiology, wrote the first book on it, really tried to understand it, and also did his experiment shirtless, either to reduce static or because he thought he looked really good. Both explanations are apparently true. Don't do that, guys. But if you do this today, it's completely reproducible. You cut yourself, there's an electrochemical current at the injury site almost immediately. And that's not understood then. That's why people kind of let it drop off the radar and really focus more on neural electricity. This has nothing to do with the brain, but it's actually really kind of prosaic. So here's your skin, any barrier tissue, epithelium, right, moves ions back and forth. One of its jobs is to separate outside and inside. That is fundamental to multicellular life, even in the simplest organisms. Now, if you break the epithelium locally, you wind up messing up the local ion gradient, but not messing up the lateral ion gradient, which means you now have a buildup of positive charge that drives towards the negative center. And this tends to be around a volt per centimeter. It really is ionic current. This is not capacitive coupling if you're an electrical engineer. This is DC current. And cells can sense this and respond to it. And one of those responses is called electrotaxis, which is the migration of a cell in an electric field. This has been detected across a bazillion different cell types. And the mechanisms are extremely confusing and complicated, but here's the simplest hand-wavy version. DC fields cannot penetrate the plasma membrane. The plasma membrane of a cell is a massive resistor. We're talking like megaohm, gigaohm level. So the field must pass over the cell. But what it can do is it can pull on and manipulate every transmembrane protein in that cell, which means that it can electrophorese proteins to one side of the cell. It can apply electrokinetic shear forces as water starts to move around. It can activate ion channels and mess with protein conformation, all of which can lead to polarity changes that drive cell motion. What it can't do is pull on the cell as a body force. So these fields are so weak, they're not going to just move the cell on their own. They're at the level of single focal adhesion type piconewton forces. So just a cartoon summary is if you put a field across this cell and these are little receptors, they all go to one side. Now, this has been measured in over 25 different cell types, many model species from Dictys to Xenopus to zebrafish to mammalian systems. It's all over the place. It's conserved in multicellular systems. Pretty cool. So it looks like a good sheepdog. And the question then would be, well, why don't we have electric band-aids today if this is so good? And there's some interesting answers to that that start to highlight things like almost every study in this field is on single cells, where there's a lot of biological data, but not a lot of sort of systems level understanding, or in vivo models where you get some actually pretty cool devices like a triboelectric generator where the articulation of these two mylar and metallized sheets produces charge that drives across a little electrode pair that can heal a mouse wound. This works. Nobody knows why it works. Can't be replicated in vitro. So it's kind of cool, but we don't have the mechanism for it, but it's really neat. There's a lot of promising stuff. These were commercial products designed for diabetic ulcers. They both folded because they didn't quite work well enough. This is a solar panel implanted in the back of a mouse to directly heal an injury. This does not work. Do not do this to yourself. So the problem is there's still a lot of ambiguity about what's good and bad because the engineering doesn't quite couple the biology to the phenomenon you're trying to control. You can't just build something and hope it works, and you can't just do single cells and hope that works. So we need some sort of intermediate system. And so what we started doing was asking, how do you do this at a collective level then? Can you do this? There's so much data on single cells, fine. Could we use this as our sheepdog? And so this was the world's smallest living dinosaur that I made as a graduate student. And I was trying to make her walk. She sort of walks, but you'll see. So what it looks like is this is the crudest visualization I can give you, but it's also the simplest. You simulate a microfluidic or macrofluidic chamber electrically. You take a Petri dish, you put down double-sided tape or silicone membrane. You put a silicone stencil in the middle of that. You pour your cells into that. You let your tissues form. You pull the stencil out. You slap a lid on the top. You pour media in. You put salt bridge electrodes. This is important. Electrochemistry is a nightmare using DC stimulation. And so this protects the system. And I can talk more about fancy designs later, but this will work. This was more precise than the standard method, which was to put lines of vacuum grease down, crack a glass slide, slap it on top, and hope you made a reproducible chamber, which is why so many papers are not reproducible in this field. Did my microphone just turn on?
Yeah.
Is anyone having trouble hearing me? Okay. So does it work? You've seen enough migration now or before to know that this is not normal. So when we stimulate this tissue, oh, come on. It starts to move. And if it doesn't, I'll do an interpretive dance. So there's the tissue, zipping along to the right. It's proportional to the strength of the field. So a stronger field increases your speed, which means you can increase epithelial migration from 15 to 80 microns per hour, which is nuts. These are slow cells, and now you're massively increasing their migration speed. The directionality is controlled by the field. And so it's kind of like a steering wheel and a gas pedal. And you can bounce fields back and forth or bounce cells back and forth. Pretty cool. How they coordinate this U-turn is its own separate story that's really elegant. There are no traffic jams. It's not like the tissue makes a U-turn by going all together like this when you switch the field polarity. Little groups decide to go this way or this way, and within 5 minutes, the whole thing's repolarized, which is ridiculously fast for a tissue like this. So again, swarm engineering is kind of cool. If you don't micromanage, you let the system kind of regulate itself, you get some cool capabilities. So how programmable is that actually though, right? You saw that with a simple uniaxial electric field, and you saw some simple cases, but can you actually use this as a tool? So this is a project we started when my lab got up and running. This was about 4 years ago, with Tom and Gowan. And we made a ridiculous device with a ridiculously bad pun name, sheepdog, that is a 2-electrode system. And so, or sorry, a 4-electrode system, 2-axis. So the way sheepdog works is you have an X-axis and a Y-axis. And inside it, you have tissue, you have these salt bridges, you have perfusing media. So it's truly a bioreactor. It's fully controlled and self-sustained, and you can produce X or Y field gradients. And it looks ridiculous and does not help the reputation of bioelectricity, but it does actually work. And so here's a population of primary mouse skin cells being told to go through a right angle, kind of cool. Here's just proof that you can analyze this, right? These are trajectories from thousands of cells, in this case, 10,000 cells all overlaid. Here's biophysical analysis showing that the orientation and order and whatever all changes. These are completely understandable, engineerable, and analyzable systems. And what's really neat then is you can move tissues just across a Petri dish. This is a whole patch of tissue moving through that maneuver now. The whole tissue is actually moving. And there is no other cue that can do this as far as we know. And what's even weirder is that the way it works is kind of like an Etch-a-Sketch. Am I too old? Do people know what Etch-a-Sketches are? Who doesn't know what an Etch-a-Sketch is? Awesome. So you got those two knobs, right? You want to make a diagonal line, you go X, Y, X, Y, X, Y. You do that here. You go X, Y, X, Y, X, Y. And if you do it fast enough, the tissue samples the average, which tells you if you're an electrical engineer, you'd say, oh, this gives me the bandwidth of the system. So the bandwidth of the system is around 2 minutes or so, that it can detect changes quite quickly. And we measure that by looking at nuclear wobble in the trajectories. So mechanically, the system appears to follow a single line with a reasonably slow detection there. And it acts like a low pass filter. So what that means is that we can make really weird things happen like this, where cells go in a circle, which is clinically useless because you don't want to do that in your wound, but this really shows how plastic migration is. And what's neat then is that basically means you just quickly change the field every 2 minutes or so, and you can produce these trajectories. You can measure the phase offset. Again, these are engineerable systems. There's nothing mysterious about it. You just define the control parameters and determine performance from that. The problem then was that the biology can get in the way. So we said, oh, we can control all of this stuff. Let's start making Band-Aids. So the first thing we did was we grew mature primary skin layers. So you mature it by adding high calcium media, and then the keratinocytes fuse, junction, flatten, and become much stronger overall. And the problem then is that you go from this nice movie on the left here, which is what we used to see, to this thing on the right. It's ripping itself apart. The edge is collapsing. If you were to look at the lamellipodia here, the whole thing is snapping. It is not good. And so the tissue is disagreeing with us. It's saying, I don't want to do that. It's like my 4-year-old. And what happens then is that it's lethal. So where you see new red occurring right at the edge, that's all cells dying during this process. This is not good, right? So you needed to figure out how to control these things more rationally. So how do we fix it? What's going on here? This was collaboration with Danelle Davenport, who provides our skin stuff, and then Gowan again, was a grad student. And what we think is happening is that the tissue is dueling, or rather collective behaviors in the tissue are fighting with external commands. And so this is claymation that I use in my outreach talks and sneak into talks whenever I can, even if it's not appropriate. And in any event, right, you tell cells to go left, but they're collective behaviors that dictate traction forces, force coupling, dynamics, cell division axes, and they disagree with this external command. So that's what we think is happening. So the idea was that this interagent coupling is messing up the sensitivity. The system is too social to control. And there's precedent for this. So here are penguins. Does anyone know how penguins don't freeze to death when you're on the outside of a penguin colony? Has anyone seen those penguin papers? They're adorable papers in physics. I think just look up penguin jamming transitions and you'll find the papers. So the way the penguins work is they're all huddled together, completely touching each other. And the penguins on the outside are going to freeze to death if they're exposed for more than an hour or so. So what happens is spontaneously, a penguin somewhere in the group pops up its head and then pushes forward by a half body length. That fluidizes the world around it, right? The crystal essentially kind of unjams, and the cells, or sorry, the penguins all take a step of half a body length forward locally, and this wave propagates through. And eventually, penguins get kicked out on one side and circulated in on the other side. The only reason the penguins survive is because they break too strong of a collective situation, and they briefly weaken collective interactions, fluidize, and then reharden later. Second example, which is really depressing, is if there were an emergency in this room and we all had to leave, there are equally useful doors, I think, I have no idea actually. People will all tend to go to the same door. And that can be modeled using a single panic parameter in a famous model from Vicek and Helbing in 2010. And it's really depressing, but it happens, right? We're overly collective. We tend to do what our neighbors do. So there are cases where you want to break collective behaviors. And so what we did, and you don't have to spend too much time looking at stuff, is just we hypothesized that it was all driven by collective cell behaviors, which were driven by E-cadherin, which was driven by more calcium in the media, which is the main thing you change for mature skin. And we showed that there is, in fact, more calcium or more E-cadherin signal in these more mature tissues. And that produces these large coordinated domains. So anything of the same color is coordinated with its neighbors. Whereas with low calcium, you get very weak coordination. Your speeds also change as a function of your calcium. So that's not good. So how do we fix that? Well, it means first that our paradigm is incorrect, that harmonizing with the existing behaviors, this nice, beautiful thing around the campfire type thing doesn't work all the time. And instead, if you can't join it, you have to actually overpower and beat the collective behavior. So what we did was use E-cadherin antibodies that block E-cadherin. It's essentially like fake hands that come in and shake hands with the proteins that cells use to bind to each other. And that weakens junctional strength because now all these little antibodies are blocking where cells would hold on to each other. It's a really nice technique for transiently weakening cell-cell adhesion. It's really expensive as well, though. But it works. So you don't get holes in the tissue anymore. You still get some pileup at the edge, but you don't get any particular damage or death in these systems now. And so if you extend that and you tweak it and you optimize the antibodies, or you use a calcium chelator very, very quickly, right at the beginning of an experiment, you go from this bad situation here to this thing here, where the whole tissue takes off across the Petri dish as a unit. It doesn't lose integrity. And when you're done with stimulation, it re-expands itself back to a normal, happy functioning tissue with cell divisions. There's a little bit of death in some compression zones, but the system is much happier overall. So that means that you can optimize the performance using drugs essentially to amplify the controllability, which allows us to do things like this, where we can accelerate healing by using a converging electric field that mimics what's found in an actual wound. We didn't try to optimize speed. We just used 1 voltage here, but you can really close things much faster now. What's interesting then is that this is pretty powerful as a cue, but if you overdo it, the cells will do whatever you tell them. They won't stop. You can drive density increases much, even beyond healing. And so our density goes way up under the electrode, which means that you can't just naively stimulate tissues. You actually have to have some feedback. Otherwise, you can overstimulate mechanically and cause some issues. So it is a really powerful cue. It completely reprograms the mechanics of these systems. So that's kind of what we see here, which is that this tissue, when it turns completely red, that's when it's stimulated. And afterwards, the center of the tissue's speed drops to almost nothing. So there's a lot of weird reprogramming of tissue collective behaviors that happens with these fields. And we need to understand more as a controllable system what that means. So the summary of that story is, if you can't join it, beat it. And we're going to pause those there and switch to something different again. How are we doing on time and interest?
About 2 minutes.
Okay, we'll do this fast. So we were very interested in working with 3D tissues because 3D tissues are obviously more physiological and have a lot of interest. And we thought maybe we could control morphogenesis this way. So we took kidneyoids, which are epithelial kidney cells, and formed these little hollow spheroids, and that's E-cadherin in purple. And we electrically stimulated them, expecting they would do some sort of branching or something that these organs or these epithelioids tend to do when you give them growth factors. And instead, what happens when we stimulate them, positive left, negative right, is they hydrostatic or they swell. So their hydrostatic pressure goes up. And that's not expected. And it goes up in a field dependent fashion. The stronger your field is, the faster it swells, to the point where it'll actually explode if you stimulate too quickly. So there's a time dependent element to this as well, which suggests some interesting mechanical properties. So we call this electro-inflation. We kind of know what's going on with it. I'll very quickly summarize that in all these tissue pressure systems, like guts, kidneys, probably lung organoids, anything where you're pumping fluids, you have chloride that gets pumped into the center of the tissue. Chloride causes sodium to follow that, and the combination of increased sodium and chloride drives water. This is controversial, but there's a lot of data in the last 3 years to suggest this is how it works, and it's mediated by a CFTR ion channel. So we use the CFTR inhibitor and we test that, and we see that we get significantly reduced swelling if we inhibit CFTR. There's some confusion here because you have to use pretty high inhibitor doses for these epithelioid models, which is normal in the organoid field for reasons we don't know, but reviewers don't like it. So we're trying to deal with that now, but it seems pretty clear that we're directly acting on the standard mechanism of inflation, and somehow we're increasing the ion concentration around this that then drives this inflation. We have a mechanically, if you weaken the cytoskeleton, you get even more enhanced swelling. So we know the cytoskeleton is providing cortical tension, which resists this swelling. And so if you knock out actomyosin contractility, everything relaxes and you can swell even more. So there are ways to really dial this in, and tissues will actually maintain that swelled state for quite some time after stimulation. But there's a lot of heterogeneity because all of these 3D culture systems are kind of a mess. So really looking in the population is a little bit tricky. If anyone's worked with organoids, it's annoying. We have a computational model that predicts this and a lot of these dynamics, but to get it right, we had to actually model the diffuse double layer around this, which I don't want to get into here because it's a nightmare of electrochemistry, but this is also controversial and reviewers don't like it, but it also is very well established in the last 3 years in a lot of papers. And things are changing. People are starting to understand the electrochemistry. And so we think we kind of know what's going on until we see something like this. So we tend to break symmetry electrically. We tend to thin the left side and not really change or thicken the right side. It tends to happen relatively frequently, and it's reversible. If you bounce the field back and forth, you can switch which side thins dynamically. And so now we have a mechanism that's more interesting for morphogenesis because you're breaking symmetry with these electric fields. And it really does hold up. The left side of the tissue definitely gets thinner over time. The right side tends not to change too much. So we kind of know this is a real process. It's really, really hard to explain. This is a field model that predicts ion concentrations around these tissues. And what we think is happening is that positive ions are driven away from the positive side and they stagnate against the cyst here. And they get taken up because this is not a dielectric. This is permeable. There's pumping. The field lines actually go into the tissue just a little bit because you have current into the tissue. You get negative ions piling up here. So we think there is ionic asymmetry. It's really hard to measure that with any known dyes, and reviewers want to see that, but we're not sure how to show that. But this is a possible explanation that might explain thinning. It could also be cell migration around the outer surface, but it's a little hard to actually prove that or see that. So there's some pretty weird stuff happening. But with organoids, it also works. And this is an intestinal organoid, even messier visually, but if you watch here, it starts to swell. If you inhibit it with CFTR at the standard doses used with organoids, again, it knocks out swelling. So we think it's a similar mechanism. And what's cool is that you get asymmetry and heterogeneity, or like localized changes based on tissue properties. So if you watch very closely, you'll see debris of dead cells in here get pushed out of the way, telling you the flow is coming in down here. This is stiffer in the crypt, which is the stem cell area. And so you start to see different parts of the tissue respond differently, which may mean there's a lot of heterogeneity just driven by geometry as well as tissue properties. But these fields can access some of this. And then the last thing is this is data with Jared Tocher's group where we took 3D mouse gastroloids and we stimulate those and they deform, which may also be mediated by gel effects with this electric field as well. But you really can access a lot of tissue programming space with these types of things. So we'll just say that electroinflation is an interesting, seemingly conserved process that we can use to poke and prod tissues. Sounds good? Okay, now we're going to change to something totally different, and then we'll wrap up with that, and that'll be the end. Okay, all right. There's a water bear. Who knows what a water bear is? Okay, water bears are the smallest animals with legs. That's not actually a controversial statement. There's only one competitor, and I can't remember its name. So for this purpose, water bears are the smallest animals with legs. They were discovered when somebody boiled water and realized this survived the boiling process. So And that's Gertz back in 1773 or so. So everyone's probably seen something like this. The New York Times likes to show this a lot. Everyone likes water bears. They look really cute. They got little articulated claws and little dustbuster noses, and they look ridiculous. And so why do people care about them besides the fact that they're cute? Here's the main reason people got excited about them. Let's compare Rasputin and a water bear. So Rasputin is famous for having been poisoned, shot, beaten, and drowned before he died. Okay, it's pretty good for a human. Water bears got all of these things and they didn't die. So they have this remarkable ability to preserve themselves in adverse environmental conditions. And what they do is they overexpress disordered proteins of 3 different types that allow them to essentially go through a phase transition where they exclude water from their bodies, fill the void space with these disordered proteins that are so good they can preserve RNA and enzyme function over time. And then when the conditions change, they rehydrate and they can reproduce again. And so people are really interested in that. We've started doing some work on vaccine stabilization with these, and Alan's doing some stuff trying to make mammalian cells more tolerant. It's really cool if you could do that. But during COVID, we couldn't do most of that. But what we did have was a lot of videos of water bears. So instead we did water bear mechanics. How do they walk? They're the smallest animal with legs. Their Reynolds number is tiny. So how are they walking? Everything else this size swims. Water bears, oddly, cannot swim. They flail around sadly and go nowhere. And they also can't even walk in traditional microscopy, which is why we think nobody has studied this before. On glass, the poor things just waggle all over the place. They can't do anything. They're not low Reynolds swimmers. But if you put them on polyacrylamide, which is 50 kilopascal jello, suddenly they take off. And they have no problem moving around. And if you image them from the side while they do that, they just look like terrestrial animals. You wouldn't know this animal lives underwater. It just has a gait that looks totally terrestrial, and it's zipping along on this gel. And you can see that the way this animal works is that it deforms the gel and actually engages with it. So this is beads. This is very poor traction force imaging, but you can see the gel deforming underneath it, and their walking speed is maximized on a higher stiffness gel, but not on glass. You need claw engagement. If we could cut their nails, we would, but we didn't have to for this study. So they're low Reynolds walkers in the sense that they latch onto the surface and generate attraction that way. They're definitely not actually swimming in that sense. And they do have a gait structure, but it kind of transitions weirdly. It's not like a horse where you have very specific gaits at specific times. Most of what they do is called a tetrapod gait, which you see in blue here, where leg activation patterns tend to result in always 4 legs on the ground at any given time. But they also will do these tripods or waves, and they even do a gallop sometimes, which is adorable. But if we were really going to go into the details, I'm going to skip this in the interest of time, but you can analyze them just like any other legged animal. You can start to understand the rules that govern them. And one thing in particular that is unique about them is the duty factor, which is how long the ratio of legs in contact with the ground versus the swing phase where you're going to the next step. Their duty factor is almost constant versus the walking speed. And the only other animal that has a duty factor like that is a stick insect, also known as the sloth of the insect world. These are lazy little critters. They just kind of walk along. And you can really just dive in and do all this cool biomechanical gait analysis to show all the different parameters of their gaits. But the main things that come out that are important is that instead of these discrete gait transitions, they tend to use a continuum of speed-dependent leg coordination rules that I'll show you in a moment. But the reason why this is important is they're tiny. Their brain probably can't handle a lot of complex dynamic processing, but they can still use these gaits. And so what actually seems to happen is that they have these interleg coordination rules, which means that the stance to swing transition, my foot coming off the ground and going forward, is going to be inhibited if my neighboring leg is already off the ground. And this probability map just basically emphasizes that a leg won't take off the ground until its neighbors already kind of back down. And that's related to other plots that would emphasize this. And I'm just going to skip over some of those data. Because the main story then is that they seem to have anatomical similarities to fruit flies called a ventral nerve cord with 3 ganglia that control each leg pair or each left-right pair, as well as these hemiganglia that handle the general pairings. And so that allows them to do this swing stance coordination without a central controller, because the legs kind of correlate their own dynamics. And that's really simple. We don't know whether they had a common ancestor with arthropods who they're not really related to genetically, or whether this was a convergent trick for small scale walking. But now that we know those rules, maybe we can make little microbots that actually walk around instead of low Reynolds swimming robots. You just need claws. Their rear legs don't follow any of these rules. They use them mostly for pushing or for grabbing when you try to knock them off the surface with a jet of water. They grab on for dear life with their back feet. And then you just need a simple controller that handles leg activity sort of probabilistically like that. So I'll stop there and thank you for your time. And the work you saw was all done by mostly these people and then these guys who graduated and survived the lab. So thank you for your time.
Biomechanics, recovery cell and the organism. Yeah, it's a big question. And
Yeah, super interesting. I was wondering, in your electro-inflation studies, you're doing ROC inhibitor and blebistatin to affect the actomyosin cytoskeleton and seeing effects. Did you also try that on the migration? Did you, yeah, electromigration?
So Yasher has done some of that work, I think, right?
Yeah.
And it's weird, right? Like you still see blebistatin, I think you still saw migration with, or,
Yeah.
and JASP, you still get a little bit with low levels of JASP, and it's like directionality and speed are decoupled, so it can still sense it, but it can't necessarily, sorry to steal your.
Yeah, so the like basically the migration speed generally decreases when you add blebistatin, like regardless of like no electric field and with electric field. The only thing that changes is directionality. And so basically from that we concluded that like electric field does not change the mechanism of cell migration within the cell, but rather changes the cell polarity or migration. Yeah, well, like I worked with like I had like 4 drugs: jasplatinonide, cytochalasin D, blebistatin, and catecholinate. I tried all of them.
Okay.
And
Yeah, I guess I was wondering, you know, like the DECMA antibody and the calcium chelation were necessary for the skin migration to work. Would there be less severe things that you could do that would still allow the collective behavior, but not, you know, have to totally disassemble the junctions?
Possibly. So one simple explanation is if you really understood the way that the field causes cells to generate force, you can use dynamic non-constant fields, right? You can pulse or use waveforms that dynamically tune things so that you don't get this massive thing. Because the one thing the body doesn't do is instantaneously generate these fields at the full strength. It takes time for them to ramp up. And so we did a bunch of experiments slowly ramping the field. And it's a little bit weird and we have to repeat some of them, but it does show that you get gentler responses that have some benefits if you tune that. But you could also imagine using a gel that has an extremely weak chelator in it briefly if you were doing therapies or things, but it's an interesting problem that is also going to change in real skin where you have a stratified epithelium. And we've been trying to stimulate stratified epithelia, but there are no good skin organoid models to do that. So if anyone has advice there.
all right, we'll take maybe in person. Looks like there might be another class. I don't know. Let's go ahead and take 2 more questions.
So just curious, I more study like cell signaling, and so I know that there are also like waves of cell signaling activity that can coordinate these like collective migrations. Is that, do you like see that the magnetic field affects signaling, or like I don't know, do you see anything about signaling that like coordinates the change in direction that you saw?
So we used an ERK kinase translocation reporter, so it's called a KTR. And it absolutely changes when the field turns on, but it also reprograms cells to move up and down instead of left and right, which means that this supposedly inert biosensor that everyone likes totally changes cell mechanics in ways nobody knows. So we can't trust it now. Like ERK changes, signaling changes, but it also, those cells are weird. So it's a good question.
Okay.
Yeah. Wonderful talk, very, very interesting and fascinating. But just two, one is two short questions. One is obviously the cell herding, I guess all the data have been shown generally from one single cell type. Have you developed strategies to assemble different cell types? That would be very interesting, possibly very useful for studying cell-cell interactions, right? It's still difficult, even using body engineering tools to
Yeah,
assemble
for sure.
different cell types.
We've tried with two cell types that move in different directions. And we can actually drive them together at an interface. And they just keep piling up there again. If you start to mix those populations, you get really weird effects that we haven't published or characterized yet. And then it just comes down to what cell type you're working with. So we've mostly been recently doing T cells and they do not respond as the literature says they should. So we're looking for fast, clean movers that are very well understood to think about those.
The other question, I think the asymmetric looming expansion, obviously you have data to show that they do have sustained effect, then the tissue becomes thinner and thinner. I think this is a wonderful technique, especially for 3D tissue culture, like organoid research.
You can tell the reviewers that.
Have you looked into maybe possible cell fate change for long term?
We would love to try that. So we haven't, we were trying to get an LGR5 mouse and we're in an MTA issue with Genentech to get the mouse. But we have a YAP mouse line that we started to look at. If you have suggestions on where to source those or a better model, that would be great. Because we can sustain the deformation for quite some time, so maybe enough to trigger signaling changes. We know that division is not changing because it's too fast for a division process, but long-term, yeah, it'd be really cool. Thank you.
We have a sheep herding problem now with sheep coming in, so thank you.